Good morning, good afternoon, good day to you all, wherever in the world you are joining us from. And welcome once again to day 5 of Aortic 2021. I'm joined by my co-host this morning, Nomonde Mbatani. Nomonde, can you believe it's already day 5 of the conference?
Yes, it's been a good 5 days, though. The content has been good, and also listening to all the international, and also hearing from other members from Africa and their experiences, and to realize how common our problems are in Africa and in some parts of the world as well. It's been a really good program.
Yes, certainly. I mean, Aortic, it never fails us. I think year after year, at all of these conferences, we certainly get a good snapshot of what is going on in Africa, but also globally. And not to mention the great opportunities for networking between all of the clinicians, the cancer scientists across the world, which brings me to the fact that please, delegates, make use of all the opportunities that are available on the virtual platform to network, to engage with your fellow colleagues, and just to drive the agenda for cancer research forward and make lives better for our patients as we move forward in terms of the cancer sciences. This morning, we really start off the session with a topic that's so apt currently on genetics, given the technological advances in modern day. We're moving into the area of big data. And certainly where genetics is really helping us to add depth to the understanding of the hallmarks of cancer, and where genetics is really helping shape translational medicine and oncology, Nomondo. So I really think that starting off today with genetics is really setting the scene for a very riveting day for us on this day 5 of our Aortic session.
Okay. Thank you, Vikash. So for this session, we've got 2 facilitators, and who are appropriate for the topic. We have Professor of Surgical Oncology, Professor Lucio Lara Santos, who's also a general surgeon in the Portuguese Institute of Cancer. And with him facilitating is also Professor Solomon Rotimi, an associate professor and director of translational research for prostate cancer transatlantic consortium. He is originally from Nigeria. Correct me if I am incorrect. Thank you and welcome.
Thank you very much, Studio, for that beautiful introduction. Yes, you are absolutely right. And I think indeed you have laid a very good foundation for this section. Once again, it's my pleasure to welcome every one of our participants to this amazing section that we have put together for you. And I welcome you on behalf of Aortic, particularly on behalf of the Aortic Research Committee, to this section. We do understand the importance of cancer, of genetics to the risk and to the whole cancer control continuum. And indeed, it's for this purpose that we have put together these sessions, an amazing panel of presenters that walk you through their discoveries and their findings as far as genetics is concerned in cancer and how it relates to African population. We know that why genetics is very important. There's a gross underrepresentation of African population in genetics and genomic studies globally. But we want to change this paradigm. And for us to change it, part of this conference, we've already had an opening pre-conference workshop. So if you missed that, I believe the on-demand recording is available for you. Please feel free to go and enjoy yourself with that workshop and educational section. This morning, as you've been told, I'll be joined also by Professor Lucio Santos, and I'll please request him in a few seconds to introduce himself as well. You are muted, Lucio.
Good morning, everybody. I'm a surgical oncologist. My interest in research is experimental pathology and treatments. And I work in Portuguese Institute of Oncology and in Angola Institute of Oncology. Thank you.
Thank you very much, Lucio. So our first presenter this morning is Dr. Luisa Pereira. Dr. Luisa Pereira is a human population geneticist and a group leader at the Institute of Research and Innovation in Health, University of Porto, Portugal. She has been using genomics to infer the past and the evolution of human population and to evaluate differential susceptibility and or resistance between human populations to complex diseases, including cancer. She has recently mapped genes that may protect sub-Saharan African population from presenting the worst clinical phenotypes, following infection with Helicobacter pylori and dengue fever viruses. Ladies and gentlemen, please welcome with me, Dr. Luisa Pereira for our presentation.
Hello, it's a pleasure to be here to share with you this talk. So I'm going to talk about how to create tools for up-to-date oncology research in the African ancestry. So since 50 years ago, in vitro models have begun to be established, and they have been very important for our acquisition of knowledge on oncobiology. So nowadays they are beginning to be developed and improved into more complex models, like for instance organoids, and of course in the more extreme scale, the in vivo models are more complex and allow us to model and mimic more complex biological systems. But even so, in vitro models are the first step in our acquisition of knowledge, but they are simple, they are easily tractable, and they allow high throughput of different testing, for instance, when testing of many different drugs, and do the first step in any selection of drug that can be used to treat a certain type of cancer. There are several panels, very extended, containing thousands of cancer cell lines, like for instance the cancer cell line is Wikipedia. Unfortunately, most of these cancer cell lines have been developed from patients that are mainly from European or East Asian ancestry. So African ancestry represents a very low proportion of the cancer cell lines available in these panels, so around 6%. And most of these African cancer cell lines were developed from patients that are from African American origin. They represent a very small fraction of all the genetic diversity that is present in the African continent. So for most of the cancer tissues that are available, there are no African cancer cell line available, or very few. So we have African cancer cell lines available for 22 different tissues. 6 tissues only have one African cancer cell line. 10 tissues have only 2. So basically, only lung, breast, and lymphoma tissues are well represented for African cancer cell lines. So this is a huge limitation on African models that can be used for in oncobiology research. If we compare, like we see in this phylogenetic tree, the huge amount of genetic diversity present in sub-Saharan African populations, with the genetic diversity represented by all the non-Africans, we see that we are missing a lot of genetic diversity that is not present in these cancer cell line models. And of course, we must improve the African representative in these models in order to be able to advance for a really an informed precision medicine agenda. So just to illustrate to you the limitation on the power of acquisition and transferability due to this limited African representative in vitro cancer cell line models, let me show you this case. We wanted to do an in vitro evaluation of a co-evolution between Helicobacter pylori and humans. As we know nowadays, Helicobacter pylori is an important factor in the development of gastric cancer. It has been a pathogen that has been inhabiting our stomachs for a long time, so more than 100,000 years ago. So by this time, human species was restricted to the African continent. So the African populations have had more time to co-evolve with the Helicobacter pylori. So in theory, it would be a longer co-evolution would lead to a lower virulence of the African stains. So African individuals would be better and naturally protected against the side effects of this infection by Helicobacter pylori. So this could explain in part the African enigma, that is, lots of African individuals are infected of Helicobacter pylori, but we don't see so high rates of cancer stomach in the African populations that as it is observed in some of the European and East Asian populations. So, of course, with the out of African migration, a small subset of individuals of African origin migrate out of Africa and gave origin to all the European and East Asian populations. And in the stomachs of these individuals, these migrants, we had Helicobacter pylori that evolved and gave origin to strains that are nowadays restricted to European populations and to East Asian populations. So this huge bottleneck that occurred with the out of African migration disrupted the coevolution that could lead to maladaptations. So potentially, as Asian and European Helicobacter pylori strains could be more pathogenic than the ones that are observed in the African populations. So we want to address this issue in vitro and try to see, to measure, to try to understand if we see this difference in pathogenicity of Helicobacter pylori in African compared with the European and Asian backgrounds. And we were quite limited in performing this study in the lab because there is only one African cancer stomach cell line available, and there are only also 2 European cancer stomach cell lines available, while most of the stomach cancer cell lines available nowadays are from East Asian origin. So we had to perform replicates of the only African human cell line available and one of the European cell lines available. So we had to do experimental replicates instead of using different cell lines that established from several individuals from African or European origins. And we infected these cell lines with Helicobacter pylori of African origin and Helicobacter pylori of European origin. We obtained the transcriptome of these assays, these infection assays that we performed in the lab. And as we can see, of course, that the human background models a lot the response that our organisms can establish against the Helicobacter pylori infection. As you can see here, the European cell lines lineages, the African ones, and the East Asians. The most interesting, we could we could identify distinct responses between the African and the European different pathways in response to the Helicobacter pylori infection. So this represents the differentiation expression of genes involved in the innate immune response. So this is the European cell line infected, the East Asian one, and the African one. The first case is non-infected, and the other two are infected by the African and the European Helicobacter pylori. And we can identify here, and I will show you attention for this case, this block of genes that are very involved in pro-inflammatory response. As you can see, they are activated when the European cell line is infected by Helicobacter pylori, but they are not activated when in the African cell line. So it seems that African individuals are more protected to not develop a very aggressive pro-inflammatory response against the Helicobacter pylori, and this may protect them against the side effects of this infection. We also could identify some signatures in oxidative stress response. So the African human cell line activated more genes that allowed the cell to respond better to the oxidative stress that is conferred by the Helicobacter pylori infection. So we could identify all these different signatures in this result, but of course we could not replicate in more individuals. So this limited a lot our main conclusions. So in order to address that, and I wanted to share with you to finalize this project that we submitted recently to funding, and we will begin it in the beginning of next year. We want to establish, with the collaboration of African colleagues, cell lines of African ancestry, and we wanted to establish them from African patients that can represent a broader genetic background of the African continent. So we will collect samples from different solid tumors, and a part of this will be collected and cryopreservated after surgery, of course, and we will try to establish cell lines from these individuals by applying a conditional reprogramming with the Rho kinase inhibitor and radiated fibroblast feeder cells. So this is a very recent technique that has a very high success rate compared with the traditional methods to establish cell lines, cancer cell lines. It's very rapid and efficient, and it maintains the phenotype and karyotype of the original sample. If this does not work, we will also try to establish organoids. But we are expected that from 100 cases, we will be able to establish about 30 to 60 cancer cell lines from individuals from the African continent. Of course, we will characterize deeply by different omics these cancer cell lines, and we will show its application by doing drug screening. We intend that these cancer cell lines are publicly available, so we will submit them after to a public repository so that everyone can have access to them and use in different oncobiology studies. So you are welcome to collaborate with us under this project, and I thank you for the attention for.
Well, thank you very much for that presentation, and our sincere apologies for the loss of audio at a particular section of the presentation. But definitely, Dr. Pereira is available on ground to provide an overview of that section. During the Q&A, and to respond to any question. Our next presenter is somebody that I like to refer to as my sister from another mother. Dr. Melissa Davis is an associate professor of cell and developmental biology, at Department of Surgery, Weill Cornell Medical College in New York. Dr. Melissa is also the scientific director of the International Center for the Study of Breast Cancer Subtypes, which is a consortium that has been studying breast cancer, well, focusing majorly on African population, but also on African American. His research interest is the intersection of race and ancestry and the influence of immunological differences in tumor biology, differences in tumoral immune response, and particularly the influence of African ancestry. Ladies and gentlemen, please join me as I welcome Dr. Melissa Davis for our presentation.
Hello, thank you so much for the invitation. First, I'll start off with a bit of an apology. It is 3 a.m. where I am, so if I am flustered a bit, I just apologize for being a little sleep deprived. I may lose my words, but happy to discuss all of what we have this morning. Let's see, so let's get started. Hopefully everyone can see my screen. Great, okay.
Oh, yes.
All right, so I was asked to speak about the pressing challenges in our oncology research, particularly for African ancestry. Here are my disclosures. I just want to mention very quickly that my research partner, Dr. Lisa Newman, who's the chief of breast oncology here at Weill Cornell, is a pivotal reason why I'm able to do all of what we do at ICS-BCS. So our vision for the consortium is basically to establish effective strategies for treatment by making novel discoveries across our very diverse patient cohort. So this is just a snapshot of all of the places across the globe where we're actively recruiting. I only have a few pictures for some of our team on the ground, but it's a very large family, if you will, of investigators who all have a common goal. Now, Dr. Newman first started making global perspective observations of triple-negative breast cancer specifically. So we know that in the United States, African Americans have a twofold higher incidence of triple-negative breast cancer, matched with a 40% higher mortality. And so as a surgeon, Dr. Newman was actively involved in clinical training and service missions to West Africa and Ghana. And she observed that the majority or a larger proportion, if you will, of patients have triple negative breast cancer in the region. And I'm going to come back to how we even knew about triple negative breast cancer in this region through the generosity of donations for resources and training to the region. And that's one key component of ICS-BCS, and that's capacity building. Okay, so one of the observations is that triple negative breast cancer was at higher frequency in the West African region compared to even other regions. And then more importantly, when we also look at other nations across the globe, we find that people who are of African descent within the subpopulations of these nations are the women who have the highest incidence of triple negative breast cancer in each one of these regions. And so basically what we hypothesize is that there's a shared genetic risk across the diaspora. And I'll just mention that, you know, aside from typical human migration patterns, the African diaspora is unlike any other human population on the planet, mainly because of the transatlantic slave trade. So unlike typical founder populations that migrate and establish hybrid populations from a population perspective, the African diaspora is one of continuous gene flow. And so it's a different model for distribution of, you know, genes in the gene pool, particularly because the transatlantic slave trade occurred across centuries and millions of Africans were displaced into the region. And so we actually see a unique imprint of African ancestry. And so strategically, where all of our sites currently reside, we're able to collect samples that represent the diaspora. And so what we aim to do is use multi-omic approaches to really investigate not only the tumor, but also the germline genetics of breast cancer patients, harnessing tools from the New York Genome Center and also in partnership with Dr. John Carpenter's lab at USC to look at not only expression profiles, but also structural changes in the genomes. And so this morning, what I wanted to do is share some lessons learned, not just about the science, because I've also talked about the science in a previous session in here in Nordic this year. But I wanted to really, show you guys sort of the inner workings of all of the moving parts. So we currently collect multiple types of samples, saliva samples for germline DNA, blood samples as well. This is for circulating biomarkers, as well as additional germline DNA. Currently in our international sites, Ghana is the only site that currently captures blood samples, but all of our sites collect saliva samples. And then we also use across the consortium, we rely primarily on formalin-fixed and paraffin-embedded tumor specimen. Now, some of our sites do have capacity to collect fresh tissue for our organoid models, which I'll get into in a moment, but also frozen tissue as well, which is ideal in genomics research. However, to have sort of harmonious protocols across all of our international sites, we stick to FFPE samples, and we've pretty much optimized this. So our ongoing challenges include, for instance, research capacity at each site. We do gap analysis of each one of our collaborating sites, and then we try to infuse training, resources, equipment, whatever is needed at each one of the sites in order to get them up to par across the board, so that we can be ensured that the integrity of the samples and therefore the integrity of the data is well enough to do comparative analysis across the sites. Of course, we have COVID-19 impact, which limits clinical capacity, restricted access to the clinics, to the lab, especially in underserved populations. And then we're also seeing altered pathology in some of the tumors. And so we're tracking whenever patients have had or are actively fighting COVID-19 in our cohort at the time of sample collections. And then one of the things we definitely chase and our goal is to bridge the gaps in knowledge. So, of course, most of the references for genomics right now are Eurocentric, and we're trying to correct that unfortunate situation, and I'll get more in detail about that, but also aggregation of populations, and I'll get into that a bit. And this certainly implies that we don't have enough information about the diversity of plasticity or, different phenotypes or heterogeneity across the disease scape. And then lastly, ongoing challenges, of course, big data handling, integration of the multi-omic processes, and then while trying to be compliant with sharing policies, also ensuring we protect our stakeholders. So as far as capacity building is concerned, our site in Ghana is primarily one of our most active sites that, we've been on the ground with training. So I'm giving you just a couple of images and snapshots of what the lab looked like when we collected our first fresh tissue specimen for organoids. So as you can see, like the centrifuge didn't have a monitor display, a digital display. However, now we've actually upgraded. So we work with Dr. Dompré in the unit that houses some of the studies for infectious disease. And we've trained a lab coordinator there to collect samples for fresh tissue. One of the things that we do because of the endemic infectious diseases in the area is also as we collect fresh blood specimens that are processed on site for separation of plasma, Buffy coat, so on and so forth, is screening for active infection before they are shipped. And logistics is something I won't get into today, but it is definitely a challenge. For each of the sites for ICS-BCS, we ideally would like to collect fresh tissue specimen for collecting or creating organoids. Right now, Ghana is one of the primary sites, but as you can see, just as an example, in our African patients, the tumors are exceptionally larger than we would see, for instance, in the United States. And so we've actually had tremendous success in collecting samples. This is just an example of one of our mission trips where I was actually training Richard Boateng, who is our lab coordinator there in Ghana. And so we work with the Englander Institute of Precision Medicine to establish our organoid lines. We have about a dozen that are, quote, growing at the moment. We have another maybe 20 that are in our freezer awaiting to be thawed. And of course, they have extensive pipelines for high throughput drug screening. We're currently working on a CRISPR-Cas9 genome assay to study the impact of certain targeted molecules on some of the African samples that show signatures that may indicate they would be treatment resistant. But also in the clinical environment. So Dr. Newman is very active at each one of our sites, conducting surgeries, but also taking part in multidisciplinary tumor boards. And we also do continuing medical education. And ideally, we would also have our teams come and visit our labs here in the US or in Ghana, where our primary site is for training in biological procedures. And this is me actually in the hood in Ghana training Richard here. And I think one of the key aspects of our success is sharing our resources. So for instance, we donate equipment. This is what I call my happy space. So it's a hood that we actually purchased and put into the lab to ensure aseptic techniques could be maintained. But then also, you know, all this luggage isn't Brittany's mini outfits. It's actually reagents that we bring directly from our lab to share and set up opportunities for our Ghanaian sites. So, you know, let's get into this lack of genomic diversity. Now, I mean, all of you already know that the primary data that is the evidence that backs up, for instance, genomic clinical testing or genetic clinical testing is underpowered for use in any other population primarily. So whether it's Hispanic, even Middle Eastern, Asian, you know, any non-European group is going to have suboptimal results on genetic testing. But there's hope. So we have new attention, not only of the African diaspora, but, you know, as our other brothers and sisters across the globe start to pay closer attention to the genomic architecture of their own populations, we're starting to see an infusion of more information that, you know, captures the current situation in terms of all of the diaspora across the globe. And I am truly excited to be in genetics research at this time, because I believe it'll certainly empower our ability to, for instance, identify African-specific susceptibility loci. So this is one of the papers that we published a few years back, and we've updated this. I think that every population could primarily, benefit from special attention to the genetic ancestry background. So here I'm showing you across our cohort genetic ancestry as we measured it. Each individual is a bar here. And as you can see for African Americans, for instance, while the majority of our ancestry comes from Africa and primarily West Africa, there's just a huge variation, if you will, across not only Africa, but even Europe and Asian populations and Native American populations in this grouping. And so aggregating this group is actually a detriment to any type of genetic analysis. And I would even, say that even European Americans across the board, you don't see as much diversity individually, but you certainly do see across the population. And so aggregating individuals by self-reported race can actually be a detriment to our ability to unravel, for instance, some of the pathways that we found that are associated with sub-African ancestry across African Americans, which we infuse with African populations.
1 minute, please.
Okay. And so I'll just close by, one of the challenges is, of course, data sharing. So we are required, because of our funding sources, to share data, but we're also very self-conscious about ensuring that we are taking care of our stakeholders. And one of the things that we are actively involved in, for instance, is creating community access gatekeepers to ensure that data is used properly for these different populations. And one of the solutions working with New York Genome Center is actually to create a cloud-based bioinformatics workflow where the data always resides in one location, and we basically grant access and the computational tools to analyze these data sets. So I'll stop there, and I'm happy to answer any questions for further discussion.
Thank you so much, Dr. Neem Davis. Thank you so much.
Thank you.
I will now pass it over to Dr. Lucio Santos to introduce our next two speakers and take it up from there.
Thank you. Professor Ananio Chaudhry is a senior scientist at the Sydney Brenner Institute of Molecular Bioscience, University of Witwatersrand, South Africa. He leads one of the HWI Gene Consortium project components and co-chair of H3 Africa Genome Analysis Working Group. He has contributed to several recent genomic studies conducted in Africa, such as the H3 Africa Genotyping Array Design, the H3 Africa Whole Genome Sequencing Study, and the Southern Africa Human Genome Program. His key interest areas are population genomics and genomics of complex threads. Please, I invite Professor Ananio Chaudhry to speak, to present your team.
Good morning. So firstly, thanks to the organizers for this opportunity to present at this prestigious forum. So today I'm going to talk about the design of the H3 Africa array, an array that is becoming one of the leading platforms for GWAS and identifying disease associations in African populations. So the array tech is based on the premise of genotyping a fixed set of predetermined genetic variants or SNPs using microarrays. So the basic protocol is quite simple. You shear the DNA, label it, and then make them bind to a set of immobilized probes. So depending on the binding associated fluorescence that you see, you can have a software tell you whether an individual is homozygous for allele A, allele B, or is heterozygous at any particular step. As these probes are often immobilized on actual computer chips, the platforms containing the probes are called genotyping chips. And there are several arrays. They come in various shapes and sizes. And the 1 million and the 2.5 million are the most standard and widely used one. And these are some of the ones that are currently very popular. So the underlying property that helps genotyping arrays capture a genome is that genetic variants in a genome oftentimes do not occur completely independent of each other, but rather as blocks and chunks. So this property is called linkage disequilibrium, or LD. The figure shows a particular stretch of genome, and you can see that due to LD, if you know what the genotypes at these three positions, which are shown by highlighting, is, you can predict what the entire region looks like in terms of variants. So the SNPs that tag LD blocks are called LD SNPs. So due to the longer evolutionary history of Africans, they have much shorter LD blocks compared to other populations such as Europeans and East Asians, as this analogy of brick walls show. So even if you sample each brick or LD block of Europeans, you would still miss a lot of bricks or LD blocks in Africans. So this makes existing genotyping arrays perform suboptimally in African populations. So coming to our array, Human Heredity and Health in Africa, or H3 Africa, is a NIH Wellcome Trust and African Academy of Science funded project to establish genomic tech and expertise on the continent. So this is a $180 million initiative spanning 34 African countries. And one of the main objectives of the consortium was to capture genetic data of about 70,000 participants across all these countries. So the genesis of our work lies in the consortium's decision to design a chip for itself. And this chip was supposed to be focused on capturing LD architecture in Africans and also come with a special price for Africa. So I will try to present this as a recipe, as this is indeed, as you will see, very similar to cooking. So the first thing in any recipe is the ingredients. So the design of an array has several ingredients. The first of them is whole genome sequences, and these need to come from genetically similar populations or, if possible, the same populations. So we sequenced around 350 whole genomes, and this added to published and unpublished data sets took us to a total of 3,000 African genomes. The second ingredient is a team of bioinformaticists. So the design was mainly done by Prof. Nikki Mulder's team at UCT and Scott and myself at WETS. The third major ingredient is computational resources that you need to store and churn these huge data sets. And almost all the work for this was done at UCT and WETS clusters, although we had some external support. Finally, you need an industry partner that would actually manufacture. A lot of discussions and deliberations, we decided to partner with Illumina to manufacture our array. And this 2.5 million array was to be given to us at an incredible price of $70 per sample. So the first step of designing an array is to generate a core set of SNPs that represents LD blocks on the genome. So we call this the scaffold. So this is more like a base of the pizza. So with our discounted price, we were given the challenge of using 1.5 million SNPs from the existing Illumina bead pools. So what are bead pools? So bead pools are packages or buckets of SNPs that Illumina uses in different arrays, and so therefore they can be ported to new arrays as a whole, and this saves a lot of design hassles. However, to incorporate this constraint, we had to generate a set of novel algorithms that selected the best combination of bead pools that suit our population. Then we use a second algorithms that added 700,000 SNPs to this so that we have a comprehensive coverage of the whole genome on the chip. So the next step was to add custom content. So as this chip was being designed with GWAS in mind, we plan to include SNPs that are known to be associated to diseases, traits, or drug response, and that are relevant to Africa. So you can imagine this as toppings. So for this, we selected all variants from databases such as ClinVar, GWAS Catalog, PharmGKB, Cosmic, and retained those that occur at least in moderate frequencies in African populations. In addition, we included 15,000 SNPs from the consortium PIs as well as other experts in the field. And then finally, we had mitochondrial Y chromosome SNPs and ancestry informative markers to enable an ancestral characterization of the populations being studied. So once this was done, we assessed how our chip was doing at each genomic region. So this plot compares the coverage of our chip, shown in blue, and the best performing competitor, and this is along the chromosome 22. And as you can see, although we do better at most genomic regions, our competitor PanAF was outperforming us in some. So to get a quantitative estimate of how we were doing, we divided the genome into 3,000 1 megabase windows and evaluated the performance for each window. So we found that around 133 windows where we were not doing as good as our competitors, and for the rest, we were either performing equally, which is shown in gray, or we were doing better. So what was the cause of this difference? The answer was simple. A bit of digging showed that the competitors just had more SNPs in these windows. So as you often finish your dish by sprinkling additional toppings to fix the lack of the coverage, we added about 100 tag SNPs to each of the problem windows. And as you can see, this fixing and patching took the orange bar out, which means that this worked. So this is what our array looked like after design. So the final step was to actually manufacture this. And although this looks straightforward, but given the complexities that are inherent to the tech, for example, many SNPs just do not work experimentally. So there were several iterations of the whole process and a lot of back and forth with Illumina, and this finally gave us our array. So the array is kind of proving to be good. It has already been used to genotype more than 70,000 Africans, and several key GWAS papers and population genetics papers, including ours published earlier this year, have been published or are very, very close to publication. So it proved to be a good array. And a version 2 that addresses some of the minor bugs is now available in the market. So with this, we also kind of learned a few lessons. So we thought there are others might benefit from this. So the first lesson is, yes, definitely you can do this here in Africa. It is difficult, but it is definitely possible. Secondly, try to get as much as data as possible before you start. Mixing and adding data at a later stage could be very problematic. Thirdly, try to secure as much legroom as possible. So when you are discussing with your partners, you need to kind of get as much as custom content, because existing beat pools are great, but they are not optimal for African populations. Finally, this is a long game. Your plan and timelines are going to be challenged. So be patient and hang on, and you will be there. So to finish the design of the array, Nikki arranged a great retreat for us here at Salmonstown, where we celebrated the long journey and hoped that the chip would provide an equally fascinating view of African genomic landscape. So I always add this to my presentation. And I need to stress that this was a teamwork, so the entire credit for this goes to the full team. Thanks to H3 Africa for trusting us with something as difficult and complex as this. And thanks to the organizers for providing us this opportunity, and all of you for your kind attention. Thank you.
Okay, thank you. Thank you for your amazing and clarifying talk. The next speaker is Professor Emanuel Gonçalves. He's graduating in informatics engineer from University of Minho and master in bioinformatics. He got the PhD in systemic biology from the University of Cambridge. He's assistant professor at Instituto Superior Técnico in Lisbon, and he worked in developing machine learning approach to integrate large-scale biological data sets to understand the proteins and metabolic regulation and their contexts. He also is a visiting researcher at the Wellcome Sanger Institute, Cambridge, UK. Please, Professor Emanuel.
Thank you so much. I'll try to share my screen. I think it was supposed to have my talk recorded. Let's see if it goes well. Can you see the talk, please? Yeah, perfect. So, well, first of all, thank you, the organizer, for the opportunity to be here and present some of the work. So most of this work, well, all of this work was carried when I was doing my postdoc at the Wellcome Trust Sanger Institute in Matthew Garnett's lab. And basically, I think it's a bit, the topic is a bit different from what has been shown in this section, but I think it really complements well. So first of all, I would like to show you some of the approaches that we have taken in Matthew's group on how to discover novel drug targets for cancer. So basically, I would like to start with the traditional drug discovery pipeline, where you basically start from a cohort of patients, and what you are really interested in is to stratify patients into the best therapy. And the way you stratify patients, the way we stratify patients, basically, or cohorts, is by using the molecular characterization of these patients. And when I say molecular characterization, this could be either genetic mutations or copy number alterations, so genetic changes, but also other molecular measurements like gene expression or protein abundance. And what I'm really interested is to take these high throughput, large scale data sets and apply machine learning and network biology approaches in a way that we can identify subsets of markers that can stratify the population based on a given phenotype. But of course, this wouldn't be either ethical or feasible to do in patients. So one proxy for that is cancer cell lines. So Matthew has over a decade now assembled a pretty large panel of cancer cell lines. These are just over 1,000 cancer cell lines covering multiple tissues and multiple cancer types. And what Matthew has also done over the years was to systematically, molecularly, and phenotypically characterize these cell lines. So we have both whole exome sequencing, for some of them whole genome sequencing, SNP6 arrays, transcriptomics with RNA-seq, and more recently proteomics. But also this provides us a molecular characterization of these lines, but also it does what Matthew has done is phenotypically characterize these lines. So every single line has been exposed to hundreds of anti-cancer drugs. And what was measured was basically the viability of these lines upon treatment. And what this tells us is which lines respond to which compound, right? And then more recently, taking advantage of the recent technology advantage of CRISPR-Cas9, what we have done was to systematically knock out every single gene in the genome of a large portion of our cell lines. So we have gene knockout viability measures as well. There is a similar effort at the Broad Institute in Boston, US. It's called the Cancer Cell Line Encyclopedia. And what the Broad and the Sanger have done was actually to join efforts to define a cancer dependency map, which has the bold goal of characterizing all vulnerabilities of all cancers. And basically, this is one of the outcomes of the analysis of some of the analysis that we perform with these data sets. What you can see here is right at the top, what you see is basically that cell lines that have HARBB2 copy number amplifications, they are more sensitive to HARBB2 knockout. Or, for instance, cell lines that have KRAS mutations, they are more sensitive to KRAS. So this makes sense. So these are very well-established cancer dependencies, and this lends credit and support to our approach. When we first published our CRISPR-Cas9 screens, what we realized was that a very strong association was coming up. That was between this Werner-Ely case that was synthetically lethal in tumors that are microsatellite unstable. So if you look here on the left, what you see is basically each bar is a cell line, and the length of the bar corresponds to how essential Werner is in this cell line. And basically you can see that pretty much among all the biggest bars, you are all golden. And these are pretty much all microsatellite unstable cell lines. So this was a synthetically lethal interaction that we came across when we were just first publishing our CRISPR-Cas9 screens. All data is published here. But so personally, one project that I led was basically to try how can we improve the drug development pipeline with these data sets. We all know that the drug development is riddled with low success rates, and part of that is due to the poor knowledge on the drug mode of action. If you look at this seminal paper from Bernard Kusta's lab, what you see is basically they have checked the affinity of several kinase inhibitors across all kinases. And we can see is that for some inhibitors, they are actually very selective, like EGFR inhibitors and MEK inhibitors, but some of them are pretty unspecific. They target, they have a high affinity for a whole family of kinases, so some of them actually across all kinases. So what we wanted to do is to compare our drug response data to our CRISPR-Cas9 data. So because our CRISPR-Cas9 data, what it provides is really a clean single gene knockout, okay? And what the drug may encompass is basically a more convoluted response of the line, because you may be targeting or inhibiting multiple proteins. So this is exactly what we have done here. So we applied a very simple linear regression model to test systematically all the possible associations between the nearly 17,000 gene knockouts versus 480 drugs that we had, and all this across 484 cancer cell lines. So this is a pretty large data set with a large number of lines. And what we have done, we look for associations exactly like this. Each cross or a dot is basically representing a cancer cell line. And the lower they are, the more sensitive they are to a drug, and the more sensitive they are to the knockout of this gene. And often when we see association like this, what this often represents is that this is the target of the drug, okay? And sometimes you may have some associations that are just proximal to the target. So this is overall what we found. What you see here in red is this drug target association. So we recapitulated actually the targets for a quarter of the drugs. And what you also see is some light gray. So these are closer to the target. So it's our proteins in the same pathway that are known regulators of the drug target. And on the other side, actually, it was interesting to see that this is Nuclein-3A and TP53. So Nuclein-3A inhibits MDM2, which negatively regulates TP53. Hence, it makes sense that the effect size is actually opposite to what we see here. So it's a negative regulation. Just to give some examples, specific examples of where this actually shed some light was, for instance, into the specificity of the compounds. We could see, we looked at PIK3C, PIK3K inhibitors, and what we saw is actually some inhibitors are isoform specific. So some of them target isoform alpha, some others target the isoform beta. And what you see here is that for those specific PIK3C alpha and PIK3C beta specific inhibitors, we recapitulated the association with the target. So these are very selective compounds then, because it mimics the CRISPR-Cas knockout. But actually, when you look to the pan-PIK3CK inhibitors, so they will be inhibiting multiple isoforms, we don't detect any significant association with targets. So this is already telling us the potential specificity of a compound, okay? And also, we could inform some potential off targets. We looked at ibrutinib. So ibrutinib is a BTK inhibitor, or BTK is the canonical target of ibrutinib. And actually, we didn't see any association with BTK. So this was a bit puzzling at first. And then we spoke with our colleagues at AstraZeneca. And basically what they told us is actually BTK covalently binds EGFR and inhibits EGFR. And that makes perfect sense from the associations that we are getting. Actually, we are getting EGFR significantly correlated with ibrutinib drug response and also RBB2. So this is basically what we see here are the effects of the off-target inhibition of ibrutinib and actually not of its canonical target. So just for the sake of time, and I also like to finish with one slide for this section. Basically, hopefully, I showed you how we've been using cancer cell lines and how we can use cancer cell lines as a way of identifying novel drug targets and biomarkers for cancer. And using machine learning approaches and network biology approaches, hopefully, we could show that how a way to process this wealth of data, because these are pretty large data sets and you actually need to process and analytically digest them quite extensively. But I think, like I said, I think it is very relevant for this session is basically when you look actually to the ancestry of these cancer cell lines, or cancer cell models in this case. Pretty much for right off the shelf, we don't have available the ancestry background for pretty much half of the models. And these are now merging both Broad and Sanger cancer cell models. And for those that we have, these are largely European, okay? So there is clearly some lack of representation and possibly there is some effort that can be applied here to actually characterize the background of these unknown cancer cell lines. And this will definitely have an impact in the end in the biomarker discovery. Either we are missing things or probably will not be certain associations in specific ancestries. I think it's also because from an earlier talk, I think there are intents in Matthew's group and so on to expand these models using organoids as well. So I think this will be a very relevant venue in the future. That's it. So I'd like to thank Matthew's group. So all this work was carried in Matthew group and while I was a postdoc in Matthew's group. And I'd like to thank the group and also specifically our collaborators for this study at EMBL, EBI, and AstraZeneca. Thank you very much. I'm happy to take questions now or in the discussion.
Thank you for your excellent talk. Please, Professor Rutami, conduct the section again, please.
Thank you very much, Eliseo. Definitely, it's been a very engaging section. It's so engaging that I can tell you we are already running out of time. We only have 2 minutes. And so I request that if you just look at the chat box, there are questions that the participants have asked. If you could respond to it, particularly just within the 2 minutes, and I will pick just 2 of them. One is to Dr. Melissa, if you could respond in 30 seconds. How do you select your site? There are those interested in.
Okay, sure. So we select them by them approaching us, or just simply their desire to want to do this type of research. We're really trying to build capacity. And so the sites that are currently active is just a reflection of those that we could activate through the material transfer agreements and IRBs and funding, of course, in place.
Yeah, thank you so much. The next question is from Dr. Pedro Fernandez, who I'm sure Dr. Anayo may know, is the co-chair for, works closely with me on the Aortic Cancer Genomics Special Interest Group. He wants to find out, the $70 price, array price available for all Africa, is it available for all African researchers? And are the companies like Illumina willing to negotiate a flat rate for African that is not affected by foreign exchange rates?
Yeah, so the first answer is, so this was the consortium price. So I think, but externally it goes for $75 or $80. So it's kind of, can you hear me though?
Yes.
Okay.
Yes.
So
yes,
the second question, I think I like this idea, although local service providers give us rates, but that's not flat. So I think that's a wonderful idea. We should perhaps negotiate with Illumina to get like a flat exchange rate. That's a wonderful idea, actually.
Okay, thank you very much to our presenters. We are now at the top of the hour. Please, all the other questions will be addressed, in the Q&A section. We'll be engaging with all the participants there. All the questions will be responded to.
Thank you very much.
And now we can now pass it over to the studio.
And thank you to the facilitator and to all the presenters for keeping up to time. Truly engaging session, and really once again demonstrating how the power of science and more so the power of interdisciplinarity and working in collaboration, large collaborations, really bringing to the fore how the power of genetics can transform translational medicine in oncology and really help us understand more about the hallmarks of cancer. So once again, thank you very much for that session.